Jinjun. So Professor Shi is an associate professor at Harvard Medical School and is a faculty member in the Center of Nanomedicine and Department of Anesthesiology at Brigham and Women's Hospital. Actually, Professor Xiaoding and I were both actually working together with Professor Shi before we came to Guangzhou about 2 years ago. And so his research involves a highly interdisciplinary combination of nanotechnology, biomaterials, drug delivery, and immunotherapy for biomedical applications in cancer, atherosclerosis, and other diseases. And his current research includes RNA nanotechnology for gene silencing, mRNA delivery for protein restoration, and nanoparticle-mediated immunotherapy. So today, Professor Shi will grace us with his lecture entitled RNA Nanomedicine, Integration of Nanotechnology, RNA Delivery, and Biology. So let's welcome him.
Well, can you hear me here?
Yes.
Okay, well, first of all, thank you so much for having me here today. And it's a great pleasure to see you, to all the friends, to see Professor John. We have been friends for many years, and Professor Xiaoding Shi, and also a new friend, Dr. Yu from Macau. So it's really a great honor to be here today. And then also, I hope everybody is well and safe because of the COVID pandemic. And that's why we have this virtual meeting here today. And actually, but to be honest, it's the same to me. And it's also my first virtual meeting to I assume a lot of audience here. So today I will go through first. Please allow me to have a couple minutes to introduce the Center for Nanomedicine at Brigham and Women's Hospital, Harvard Medical School. We are in the Boston area, and this is if you came here Boston before, or you're familiar with Boston, and then you like soccer, like baseball, and then we are in the Fenway area, and then we have the this area for Red Sox team. And this is our kind of new building. It's been like almost 4 or 5 years old. And then we are on the 7th floor of this called the Hill building for transformative medicine. So that the aim of this building is to really develop transformative materials and medicines for patients. And this is a mixture building either with the clinic and the research. So the center was actually established by also my mentor, Dr. Omid Faloxad, and also actually the mentor for Phei Er and then Xiaoding. So now we have 3 labs, and Dr. Faloxad's lab, which focus on, as you know, he has done a lot of work with the nanomedicine and a lot of translation work he has done, including biotherapeutics, and also he's very interesting recently on nanobio interactions, and has another company called Seer, and to work utilize the nanobio interactions to study the proteomics. And also we have another lab here. It's called Dr. Jeffrey Cobb's lab, and he's actually not in our department, but in the Department of Medicine, and his lab is focused a lot on the biomimic materials like adhesives, devices. Also, he works with stem cell engineering, and he also has started probably 6-7 companies, and one of them went to public a year ago, and then the other one you probably heard about that is called a Gecko, and to put the adhesive gel on the as adhesive materials to heal the injury, or the surgery after surgery of the heart, and it's actually in phase 2 clinical trials. And also, the center has I started my lab about 7 years ago, and our research is focused on mobile responsive nanotechnology, and for particularly biomicromolecule delivery like siRNA or messenger RNA. And we're also certainly interested in immunotherapy, which is hard these days because of the success of PD-1, PD-L1, and then CAR T cells. Also, we're gonna expand this center, and we'll probably soon have another new lab joining the center. So today we'll briefly go through some of our research, but before that, let me first of all introduce overview of what we are doing. And as I mentioned, we are very interested in developing, well, not entirely new, but more rational materials in nanotechnology. and which hopefully we can solve a lot of challenging issues in the systemic delivery of biomicromolecules such as small interfering RNA, messenger RNA, and protein. That's why our main interest in the lab here. We like big molecules instead of small molecules. And so hopefully my talk is somehow very different from Professor Zhang's talk, which is fantastic. And our biomedical application is also broad, but the main focus is on the cancer and atherosclerosis. And more recently, we are also interested in something else like pain medicine too. And meanwhile, we're thinking fundamental research is critical for our design of biomaterials and nanoparticle. So we certainly would like to understand more about how nanoparticle interact with different biological systems, such as serum protein, because this is the first step, right? When you inject a nanoparticle through IV intravenous injection, the first thing nanoparticle nanomedicine met or sees is the serum protein. So this interaction is critical, and how this interaction interact with the pharmacokinetics, biodistribution, efficacy, penetration, etc., all critical, right? So we think the fundamental understanding of the nanobio interaction will help us to develop a more rational or more, effective technology or materials. But because for the sake of time today, I won't have much time to talk about the fundamental studies. We'll mainly focus on today's talk on the recent work on developing RNA technologies, including both siRNA and messenger RNA, for treatment of cancer and atherosclerosis. I'll give you 3 examples. So let me start first with cancer. So cancer, as we know, is a very complex disease. Everybody knows that it's hard to treat. It's always come back, even you initially have a successive response. And majority of cancers we know involved with the genetic mutations. Compared, if you look at these ACR figure here, compared with normal cells, as you can see, the normal cells, everything's balanced in, between the oncogene and tumor suppressor. But when this balance is broken, you have the overexpression of oncogene or more broad, the pro-tumor genetic genes, and then the loss of function of tumor suppressor, either through mutation or complete deletion. And then this balance is broken. Then the normal cells become tumorigenic. It becomes the tumor cells, and then later on it will metastasize to other places. Also, these genetic mutations can cause the drug resistance. So that's why in many cases the initial treatment was successful, but later on the cancer relapsed. So in current, I would see majority of pharmaceutical companies, they are focusing on the targeting of pro-tumor genetic drivers, and many drugs have been successful, including more recently the precision drugs. But unfortunately, still there are a lot of pro-tumor genetic oncogenes. They are considered as undruggable or difficult to drug. Either it's hard to design small molecules to target these therapeutic potentially proteins, such as the transcription factors. And so that's why one thing we think RNA interference technology could be very helpful to potentially to target those difficult or undruggable proteins. And on the other hand, tumor suppressors, in many cases for the cancer, they are either depleted completely or mutated. So there are very limited strategies we can do to tumor suppressor. There are certainly some small molecule development to target the upstream or downstream pathways. But if you want to completely restore the tumor suppressors, certainly there are very limited options. And unfortunately, so far, I don't see any FDA approved or clinically approved therapeutics to target tumor suppressors. So that's why we think using messenger RNA could potentially provide an option for this lost tumor suppressor function. So let me start with the first technology I would like to introduce to you. It's called RNA interference technology, RNAi. And I'm sure many of you know this technology. It's been a while. First discovered in 1998 by 2 scientists, Andrew Fire and Mellon. And one of them actually is in the Boston area, Craig Mellon in the UMass Medical School. It's about a 1 hour driving from Boston. And as you know, on the left side, we can see this is a simple schematic of how RNA technology work. Either the long strand, double strand RNA in the tumor cells can be degraded by the dicer enzyme into the small interferon RNA, and the small interferon RNA integrated to this ARGO2 RISC, because you got RNAi silencing complex. This is protein complex together, and then the sense strand will be cleaved, and the antisense strand together with this RNA assembly will target specifically the mRNA. So depending on the sequence, you can almost target any gene you want. So it's a very potent technology, and when it was discovered, particularly after in 2001, after the report of synthetic siRNA. So many pharmaceutical companies have had a lot of enthusiasm for this technology, because you can specifically target the gene and then inhibit the protein expression. So you can target almost anything you want. And then right after that, 5 years later, they both, Andrew and Craig, won the Nobel Prize in physiology and medicine. And then it took 12 years, another 12 years. So in total, you can see from 1998 to 2018, 20 years exactly. We saw the first FDA-approved siRNA therapy for a rare genetic disease called HATTR amyloidosis, which was developed actually here in Cambridge, in Massachusetts, by a company called Alnylam. So it took like 20 years from the discovery to get the first FDA-approved drug. It's reasonably long time, but it's also a reasonably good amount of time. The reason is because small interferon RNA has some intrinsic features, such as large biomolecules. Its size is certainly much larger than small molecules. And also it's because it's negatively charged and highly hydrated. So it's really challenging for this siRNA to cross the cell membrane, which is also negatively charged in many cases, to cross the cell membrane and into the cytosol. And also the siRNA, as we know, could be easily degraded by nucleus. But I would like to point out here, this is probably, the third point is probably not the big issue anymore these days, because of the advance of a lot of useful chemical modification strategy to stabilize the siRNA or other nucleus. And the fourth one, the challenge is actually, or the features for siRNA delivery is we have to make sure we transport the siRNA into the cytosol, where this RNAi silencing strategy, pathway worked. So I think, here we go. So over the last 10 years, since 2010, when I was a postdoc in Omi's lab, and then we have been developing different nanoparticle technology to trying to solve the systemic siRNA delivery to the solid tumor in particular. So we've been working for many years and then develop a variety of different strategies, including some strategy developed by Professor Jinjun Shi. And the aim of our platform technology are actually to solve many challenges in the systemic siRNA delivery, including long broad secretion. How can we improve the broad secretion? As we know, a small siRNA by itself is very, can very quickly degraded by, in the blood. It takes probably like 30 minutes. It's like small molecules. And also the second challenge is how can we improve the tumor accumulation? And the third one is actually is far more important, and then we gradually realize it's critical for the cancer nanomedicine field is how this nanoparticle can penetrate through the tumor tissues to reach out to as many tumor cells as possible. And then also we certainly want more, cell uptake, and particular specific cell uptake by either tumor cells or other tumor cells in the tumor tissues. And then endosomal escape is another critical thing, and then that's how nanoparticle can help. And certainly control release, how can we get a sustained silencing is also another challenge. So we have developed this technology to solve, I would say, some of these issues at one time, but not yet, as to my understanding, is to solve all these problems at once. But we're, getting very close to this, and then hopefully next time I can report to you I can share with you new technology we're developing now to solve all these problems at the same time. So I will just give you one example to how this technology can be used for cancer treatment. So we've been collaborating with Dr. Bruce Zeta from Children's Hospital in Boston. And his laboratory discovered an interesting protein called proheptin-1. And in the clinical sample analysis, they found in later stage of non-small cell lung cancer, high expression of PHB1 is highly correlated with the low overall survival of non-small cell lung cancer patient. So, but unfortunately, there are no specific small molecule inhibitors that we can use to target the proheptin-1. So when I talk with Dr. Zeta, we think the RNA technology is perfectly fit to evaluate these targets, say whether this PHB1 target could be potentially used for cancer treatment. So we worked together for many years already. And initially, we designed the nanoparticle called the lipid polymer hybrid nanoparticle system. Later on, I'll explain a little bit more about the platform for RNA delivery. And with the control nanoparticles, as you can see here, so you don't get any silencing. By the way, the red staining is the PHB1. And with the nanoparticle loaded with siRNA PHB1, you can see almost complete silencing of the target. And with the silencing, of course, you can see the apoptosis and very clear apoptosis after the nanoparticle siPHB1 nanoparticle treatment. And when we do the in vivo intravenous injection, we can see the nanoparticle itself is pretty potent by inhibiting the tumor growth. Also, we can combine this nanoparticle with small interferon PHB1 together with platinum drug, which is the most widely used chemo drug for cancer treatment, including for the non-small cell lung cancer. So we can even get an even better on the human anti-tumor effect. So that's one example, and we have been working on this for many, and collaborating with many other scientists and physicians in this field. So hopefully we'll get something more meaningful done soon. And then I will move to the second topic, the second technology, which is messenger RNA technology for protein restoration, protein replacement. Well, you know probably a lot about messenger RNA now and heard about all the time because of COVID. And then again, another company in the Cambridge side called Moderna developed the messenger RNA COVID vaccine, and which is I think it's about late last week. They moved that to the phase 3 clinical trial. So hopefully we will have successful COVID mRNA vaccine soon, and to help the patient, and to hopefully can solve the pandemic issue. So compared with DNA, both messenger RNA and DNA can be used for protein expression, right? So but mRNA is certainly in the middle, and you do the DNA transcription to mRNA and mRNA translation to protein. So compared with gene therapy, which is based on DNA, we think using messenger RNA has several benefits. On the right side, as you can see, first of all, there's no nuclear barrier, so you just need to deliver mRNA to cytosol, not necessarily to the nuclear, like what we have to do for DNA. And also, you can, because of that, then you can avoid the host genome integration, so you avoid any potential toxicity issues. And the mRNA-mediated protein expression is faster and more predictable compared with DNA. Also, because of the robust chemical modification, now we can see have a very we can have the synthetic messenger RNA with much reduced the immune response and better stability and transfection efficiency. But overall, similar to siRNA, and the key challenge is still how can we deliver the messenger RNA into the cytosol. So the cytosolic delivery is certainly a big challenge. And that's why a lot of different strategies, particularly in lipid nanoparticle technology, have been developed to solve this potential issue. And so in our lab, we started working with messenger RNA about 6-7 years ago. Indeed, we first, when Moderna started the company a couple of years later on, we realized this field could be, very interesting, and then could solve a lot of potential problems. So that's why we started working on this field. And then we certainly did not develop, entirely new nanoparticles for the mRNA delivery. Instead, we have done a lot with the the lipid polymer hybrid nanoparticle system for siRNA, and then we transform the technology for messenger RNA delivery. So this is a very simple formulation steps. First of all, we've complex this messenger RNA with a cationic lipid-like materials. We call it here the GCOC14, which is the generation 0 of PAMAN, polyamidoamine, and we modified it with the hydrocarbon tails, 14 carbon tails. And then this is lipophilic lipid-like materials, then complex with mRNA through the charging interaction. Also in the organic solution, we did dissolve the polymer called the polylactic lactic acid, PLGA. And then we added this DMF solution into water. And then because of the DMF solution is miscible with the water, so the polymer PLGA, which is hydrophobic, and then we'll precipitate it out and together with this complex. And as you can imagine, this mRNA GCOC14 complex, they sort of like have this structure, which we don't know yet whether it's really is a reverse micelle structure or not, but then through these lipophilic or more hydrophobic tails interaction with PLGA, this complex can be trapped in this PLGA core. And on the surface, we also dissolve this lipid pack on the in the solution, in water solution, and then this lipid pack can then self-assemble on the nanoparticles to stabilize the nanoparticle, and also to that also give the nanoparticles long circulation in the blood. So we did a lot of characterization of the nanoparticles, and you can see the size of all these mRNA nanoparticles. It's about 100 to 120 nanometer, and it's quite stable in different mediums, including in serum. And we tested the toxicity of the nanoparticle, particularly, most of the concentrations we tested, we don't see obvious toxicity. And then also we compared with a commercially available transfection agent called lipofectamine 2000. I believe it is L2K mRNA. And then mRNA nanoparticles show comparable, even better in high concentration with the in terms of transfection efficiency in the tumor cells. So with this platform, we think what we're going to do next, right? So and then when we started this project, first we wanted to actually do vaccines, but then we realized there's a very interesting niche people have not realized a lot. It's the I talked about it earlier, the tumor suppressor loss of function issues in the tumor in cancers. So first the target we pick it up is called P10. Why in the prostate cancer? So why we're interested in this is because in later stage prostate cancer patients, there's about 40 to 50% patients, they have this loss of the P10 function. So we think this is going to be great for as a proof of concept study. So we use these P10 mRNA nanoparticles to transfect the P10 non-prostate cancer cells like PC3 cells. So you can see here this green indicated, which is actually hemagglutinin, but it's tagged to P10. So you see the green means you see the P10 expression. Very, very nice expression by our nanoparticles, and then that can induce actually very good apoptosis of the PC3 cells. And we did the Western blot, and you can see the main biological pathway of the P10 resolution is the inhibition of a PI3K-AKT pathway. And in this case, we can see the dose-dependent toxicity of P10 mRNA nanoparticles. Also, we did another P10 loss to prostate cancer cell line called LINCAP. Similarly, we can see this effect. And very interestingly, I didn't put the data here, is if you look at the P10 expressing or P10 normal wild type prostate cancer cells or normal cells, you don't see much toxicity at all after the P10 transfection. And then we move on to study whether this nanoparticle can be used to treat the tumor in vivo. And we established a model called the intratibial tumor model. The reason is because in about 80 to 90% of later stage prostate cancer, they have bone metastasis. So that's why we're interested in the bone tumor, in the tumor in the bone tissue, see whether we can deliver the mRNA nanoparticles to there. So we did the injected the PC3 cells, which is luciferase expressing PC3 cells, into the tibia, and then we monitor the tumor growth using IVS imaging system. As you can see, after treatment of nanoparticles, we do see the inhibition of the luminescence signal as compared with the control groups. So we are pretty happy with this, and we are actually also working these PTMI nanoparticle in combination with other therapies. And the next target we are interested in is actually P53, which is probably more popular, even probably is the most popular tumor suppressor in the area, in the field. And then we want to see whether the restoration of tumor suppressor, particularly like P53 here in this case, whether we can improve the efficacy or sensitize other therapeutics like chemotherapeutics. So we pick up this called mTOR inhibitor everolimus. Everolimus is an FDA-approved drug indeed for certain type of cancers, but not for other popular, more widely, distributed cancers like lung cancer, like liver cancer. Certainly, the failure of the clinical trial of everolimus in these type of cancers certainly is very complicated, and lots of biological studies have been done in the field. And one potential biological mechanism underlying the failure of the everolimus or underlying the heterogeneous response of everolimus to different cancers is because of the activation of autophagy. And then the autophagy activation by the N2 inhibitor can induce a protection of the tumor cells. And then particularly in the P53 lost cancer cells, we also see these anti-apoptosis, also see the cell proliferation, cell cycle loss of control. So all these issues are associated with the P53 lost tumor cells. So very interestingly, we all know P53 has a lot of functions, right? And one particular function is they can induce the apoptosis, they can induce the cell cycle rest. This is all well known, but also there's another unique feature of P53 in the cytosol, not the nucleus P53, but the cytosolic P53, that can actually, overexpressing that P53 can inhibit the autophagy pathway activation. So we think if we can combine P53 nanoparticles together with N2 inhibitor, we probably can see a very potent anti-tumor effect. So that's what we did this, with the hypothesis we did the test of the P53 mRNA nanoparticles together with everolimus in P53 lost tumor cell, particularly lung cancer cells and liver cancer cells. So we can see here in this Western blot data, if you can see with only everolimus itself, it actually inhibits the mTOR, as we can see, the phosphorylation. Thus induce the autophagy activation, looking at BACKLIN1 and MCLT3-2 expression. If we just look at the lane 2, the P53 mRNA nanoparticle itself, we can see it doesn't affect these autophagy pathway, but here, and because it's not activated, but it does induce the anti-tumor, anti-apoptosis. You can see the downregulation of the BCL2 pathway, and then the inducing apoptosis. But if you combine these two together, you can see here very clearly, it does inhibit the activation of the autophagy pathway. You can see here the last obvious band of the BACKLIN1 and the LC32. And then also, meanwhile, it keeps the anti-apoptosis effect. You can see the downregulation of the BCL2. But then at the synthetic effect, we can see more apoptosis from the tumor cells. So on the right side, you can see the combination did induce much higher cell toxicity and then much higher apoptosis. So we did this in cancer, liver cancer and lung cancer models. So this is just one piece of data I'd like to show you here. By the way, this drug is everolimus, is an auto drug, so auto available. So you can give auto gavage into mouse, and then, but by itself, it doesn't induce much effect, anti-tumor effect. And for our p53 mRNA nanoparticle, we give the IV injection, and you can see here that does induce the inhibition, similar to what we saw in P10 study. But when you combine these two together, you can see much better anti-tumor effect. And because of a time limit, I think I need to quickly move to the last topic I would like to share with you, atherosclerosis. As you can see, cancer and atherosclerosis have actually a lot of similarities, because for number one, both tissues are leaky. So you can see cancer is leaky because of inflammation. And again, similarly to atherosclerosis, also because of inflammation. And then the endothelial cells on the plaque tissue of the atherosclerosis is also leaky. So nanoparticle can certainly passively cross these leaky gaps and enter into the diseased tissues. And another similarity I would see is also for atherosclerosis, genetic variation highly involved. And one type of one cell type is very critical in atherosclerosis, is called a macrophage. So the macrophages, the macrophages cells, they're supposed to do is to actually enter into these plaque tissues. They eat these dead cells, the small necrotic core, and then supposedly they to reduce the core area. So this is a process called aphelocytosis. But instead, in reality, the macrophage cells did actually the opposite way. So they did not eat much of these debris cells or the necrotic cells. Instead, they actually become more inflammatory. So they induce more inflammation and check more macrophage cells on other immune cells into the plaque tissues, and then increase the plaque size, and then eventually it will reduce the rupture of the plaque, and then induce a thrombosis and heart attack, as is the end of story. So what we think, with using siRNA nanoparticles could potentially to target the macrophage, and then they inhibit the specific pathways in lesional macrophage, particularly the inflammatory pathways. So one pathway, our collaborator Dr. Eric Tabase from Columbia University, his lab discovered the pathway called the CAMK2 gamma pathway. It's a calcium modeling kinase 2 gamma pathway. And in human tissues and mouse tissues, his lab found, in both cases for atherosclerosis, the CAMK2 gamma protein are highly more overexpressed. So we think if we can target the CAMK2 gamma using siRNA nanoparticles, or whether we can induce any effect, therapeutic effect for atherosclerosis. So that's why we designed a very similar nanoparticles, and using, again, this is called lipid polymer hybrid nanoparticles. You can see we don't change much of the components. Again, you use the same cationic lipid-like materials. We're also using a DSP pack here. The difference here, we added the specific ligand called the stabilin 2 peptide to the DSP pack, which you can specifically target lesional macrophage. And also we have this POGA core in the middle, and then the siRNA trapped inside of the nanoparticles. So we did a in vitro, in vivo testing to see whether these S2P peptide targeting nanoparticles can have more macrophage recognition. So that's what we did here in vitro studies. You can see, yes, when we increase the density of the S2P ligand on the nanoparticle surface, we did get better cellular uptake or more positive staining of the macrophage. Also, we did in vivo studies compare with non-targeted nanoparticles to targeted nanoparticles. We can see about twofold increasing of the accumulation of nanoparticles in the atherosclerotic tissue, and particularly to the macrophage cells. So then we want to see whether this CAMK2 gamma could be silenced, and then whether the silencing can induce the reduction of necrotic core area. So that's what the result we got, and pretty positive. You can see here after few injections of the siRNA gamma nanoparticles by intravenous injection, we can get about like 60% silencing of this targeted protein. That can also induce the overexpression of a downstream protein called the MERTK, which is actually a good protein for atherosclerosis. As you can see here is a tissue staining of the CAMK2 gamma in the macrophage cells. You can see here, similarly, we can get a 60 to 70% silencing of the protein expression. And this actually led to the the reduction of the necrotic core area, as you can see here. This is an area size, and this is two control groups. So, and then certainly we have a lot of other data. So if you're interested, you can take a look at our just recently published a paper in Science Translational Medicine. With this, I would think I will probably end my talk. Sorry, I probably took a little bit more time than I supposed to, but I would like to emphasize here is we all know research for any area, there's a research gaps, and then it's very important, particularly for, for similarly, I would say, to RNA medicine field, that we first of all need to figure out what disease and cell type we want to target, and then what biological proteins you want to target. And then also we need to understand, what the delivery challenging is for the RNA delivery to the disease or to a cell type. And certainly nanotechnology or other technologies, I would say more broadly. And how can we use these technologies to solve the delivery issues, to solve the, the challenging the delivery barrier. So, and then we can design a successful product, and move these forward to clinical studies, right? And also we certainly need to consider the formulation, scale up, manufacturing, toxicity of all these technologies. But long and short is we all think, integration is highly important. And I think, this journal biointegration, Phei Er, and then her colleague started, is a very good platform for these, multidisciplinary studies. So certainly I've already, I assume, I would expect that this journal will be highly impactful soon. With this, I'd like to thank, my collaborators, particularly Dr. Eric Tabase for the atherosclerosis work, Bruce Zeta for the siRNA and P10 work, and Dan and the other colleagues for the P53 work, and then also the team here. They contribute a lot to these studies and the funding support. I would like to thank you very much for listening to my presentation, and I wish you're safe and health in this pandemic period of time. Thank you.
Oh, thank you so much, Jinjun, for that very wonderful talk. So as a person who works in nano, I cannot emphasize enough on the importance of what Professor Shi has just said, because, like RNA and RNA-based medicine is actually, really, the problem is they are not stable. So if you have something that can prolong the stability from 5 minutes to 30 minutes to 1 hour to 5 hours, I mean, that's good enough to being translated into clinics. So thank you, Jinjun, for that